This will be a review of the key concepts and brain function for our unit exam. We've learned that the surface of the brain is called the cerebral cortex, and it appears to be functionally specialized. Different regions are performing different jobs. For example, the back of the brain, the visual cortex, is visual cortex because it's receiving inputs from the eyes. In the temporal lobe is the auditory cortex. It is receiving inputs from the ears, etc. So the nature of the inputs to a brain region determines in part what that brain region is going to be doing, what kinds of information processing is happening at that location. Likewise, the hippocampus. Underneath the temporal cortex is the hippocampus, and it receives inputs from all the major sensory perceptual regions of the brain, so it can make a rich episodic memory trace of what's happening at any given moment. And so too the amygdala has certain inputs. It receives sensory inputs, and in fact it is the location of the memory trace for simple associations like a tone and a shock. Neurons communicate with electrical signals. Here we see a neuron. It has dendrites, the receiving zone of the neuron. The cell body is going to integrate the electrical activity received at the dendrites, and then the axon will carry the output electrical signal down to the axon terminals where chemicals are released, and these chemicals can electrically excite the target neuron. In this picture then we see synapses, 1, 2, 3, 4, 5, 6 synapses, the junction between an axon terminal and a dendrite. It is here where small electrical signals are generated, and then the cell body will, in a sense, add up all these small signals. If they reach a threshold, the cell body will generate its own output signal that will race down the axon to the terminal, and chemicals released. So electrical signals are the language of the brain. And neurons adjust the frequency of their electrical signals depending upon how much activity it is receiving on its dendrites. So if all of the synapses are currently active and we're getting all these small electrical signals, it'll generate a large electrical signal. In fact, the cell will produce a high frequency set of electrical signals if all of the synapses are active at the same time. However, if only 1 or 2 out of the 6 are active, the cell will generate just low frequency electrical signals. And so the frequency of signals carries information. If this were a sensory neuron, for example, the low frequency signals would indicate a soft sound or a soft touch or dim light. High frequency signals would indicate loud sound, hard touch, or bright light. In general, though, the frequency of the electrical signals is dependent on how much activity it is receiving on its dendrites. The function of brain regions can be studied from different perspectives using different techniques. Let's take the face area in the brain. I'm going to start on the right here. We know that damage to a certain area of the brain in the fusiform gyrus on the right hemisphere, if damage to that area of the brain produces face blindness. People have difficulty perceiving faces, the identities of individuals. They can't seem to remember the face or identify them later or even imagine the faces. Stimulation of that face area causes distortions in face perception. This was an epileptic patient who had an electrode that happened to be right over the face area, so they stimulated that area while he was looking at the doctor, and the doctor's face got distorted. The fMRI scanner can measure the activity, the functional activity of different brain regions, and the face area becomes highly active when the person's seeing a face compared to seeing an object. Face, object. And this signal would be indicating the change in blood flow in that region. In fact, fMRI then is a technique to measure the functional activity of a brain region, because when a brain region becomes active, it recruits a larger blood supply. So here we see the brain. It's full of blood vessels. Every brain cell, every neuron is somewhere close to a blood vessel. Blood vessel brings food and oxygen to the brain cells. Here we see in this picture, if these two neurons were to become active because they're processing information, they will signal the local blood supply to increase the flow of blood. So the blood vessels bring more food and oxygen, and that blood flow change is detected by the fMRI machine. So fMRI does not measure brain cell activity directly. It's measuring blood flow as a substitute for brain cell activity. Now, neurons have a preferred stimuli. We've learned that in the visual cortex, neurons there have preferred stimuli of edges of different orientations. In the face area, we suspect that these cells in the face area will have a preferred stimulus of a face. And we've even learned of a neuron in the hippocampus, again from an epileptic patient who had an electrode inserted in this region. They found a hippocampal cell that responded to a video of The Simpsons. And that cell responded just to The Simpsons video and not other videos. So neurons have a preferred stimulus. We also learn about the psychology of learning. In these 4 pictures, we see the process of teaching a dog to salivate to the sound of a bell. The food is the unconditioned stimulus, and the salivation to the food is the unconditioned response. And this happens automatically. The dog's nervous system is wired this way. The bell would be the conditioned stimulus, and notice at first it's not generating a response in the dog. But in picture 3, we're going to pair the bell with the food, bell, food, repeatedly. Now, initially, the dog is salivating because of the presentation of the food, but over time, the association of the bell with food will lead the dog to salivate at the bell alone. This is the conditioned response. Likewise, in fear conditioning, the tone would be the conditioned stimulus, and then the training would be play the tone and the shock. The shock is the unconditioned stimulus, and the rat naturally fears the shock. So that would be the unconditioned response, the fear to the shock. But if you pair tone, shock, pretty soon you can play the tone alone and the rat will show fear. That's the conditioned response. Now, neuroplasticity allows for learning and memory and the updating and erasing of memories. So we learned early on about Clive and H.M. that the hippocampus was a critical structure in making new memories. Without a hippocampus, they couldn't make episodic memories. And so scientists discovered that there's lots of neuroplasticity going on in the hippocampus, and this allows the hippocampus to make episodic memory traces. In fact, there's neuroplasticity in the cortex as well, but we said that during memory consolidation, the hippocampus is needed to help to strengthen those cortical memory traces. But again, the whole surface of the brain is prepared for neuroplasticity as we learn things. In the Memory Hacker movie, we learned about the sea snail, that when it was touched and then shocked, touch, shock. And this was Eric Kandel's research, and he discovered that what's happening between the sensory neuron and the motor neuron is that new synapses are growing, and this is the basis of long-term memory. So the memory trace is the physical change that happens as a result of learning, in this case the growth of new connections. This means then that the touch alone will generate that response in the sea snail. It has learned to associate the touch and the shock. Likewise, for the dog condition, the bell and the food, we can think of the 2 neural pathways as converging on the common target cell, the blue cell. When the blue cell gets turned on, the dog will salivate. Well, before training, the bell does not activate the target neuron. It's a weak synapse. The food will activate the target neuron. Strong synapses, many synapses here. So before training, only one of the pathways can activate the target cell and cause salivation, the US pathway. But after training, after bell, food, then what we have is neuroplasticity. We have the growth of new synapses in the bell pathway. As a consequence, both pathways can generate salivation. But when it's happening by the bell alone, we call it the conditioned response. We also learned that memories can be erased. So we saw this woman here who had a fear of spiders, and what the researchers did was they had her retrieve the memory, so interact with the spider to retrieve the fear memory, and then take a drug that blocks reconsolidation. And she and others experienced sort of a significant weakening to the point of erasure of the memory. We know the amygdala is a critical component in emotional memories, and we've used the amygdala to illustrate what erasing a memory might look like. So here we see the tone shock pathways converging in the amygdala. If we pair tone shock, we get the new synapses in the tone pathway. So this is the memory trace for the fear memory. But if we have the animal retrieve the memory, we play the tone alone and give it those drugs, we can effectively weaken those synapses again, so that we're erasing the memory. There happens to be another way to do this, and that is to use an extinction procedure as well. So you have the animal retrieve the memory or the person retrieve the memory, and then you present the conditioned stimulus by itself. So in this case, the tone alone, tone alone, tone alone, it seems to have the same effect of weakening the original memory, erasing it. Now, neuroplasticity tunes up neurons to have a preferred stimulus, both during brain development and subsequent learning. We learned that if we put goggles on a kitten, where the goggles have only horizontal edges, later on as a cat, they have lots of horizontal edge detecting cells, but they have very few neurons that can respond to these other orientations. Very few vertical edge detecting cells. As a consequence, they will be blind to vertical edges. We also learned about the hippocampal neuron that responds to The Simpsons. Now, it's not like the person was born with such a neuron. Rather, for any learning experience, there will be some hippocampal neurons that will respond to that event. And then those hippocampal neurons will undergo synaptic plasticity with other hippocampal neurons to form a memory trace of that episode. So they become Simpsons neurons as a result of the learning episode. Likewise with the tiger. When we see the tiger, we are activating a certain set of neurons in the visual cortex and auditory cortex if the tiger is roaring at that time. Now, we weren't born with tiger neurons, but the activity here is going to create tiger neurons so that in the future, these neurons then will be reactivated when we are thinking about tigers or imagining tigers, or we recognize a tiger. So neuroplasticity is what tunes up neurons to have a preferred stimulus. Memory consolidation transforms a short-term memory to a long-term memory, and an episodic memory to a semantic memory, and the hippocampus is a key player. So we learned with those neurological patients like HM and Clive, if the hippocampus is either removed or damaged, then a person cannot make new episodic memories, because this is the location where all that information is being tied together, linked together in the hippocampus. But if you do have a functioning hippocampus, then what happens is the hippocampus, over time, especially during sleep, the hippocampus will help to strengthen the cortical memory traces. And this process is called consolidation. As a result of consolidation, we have strong memory traces in the cortex that can be reactivated without the hippocampus. So this will be semantic memory stored in the cortex. But again, if you lose the hippocampus, you will lose the rich episodic memories. And this was our diagram in the tiger situation. After seeing a tiger, we go home, we go to sleep. The hippocampus will be signaling the cortex to strengthen those cortical memory traces involving the tiger experience. Memory retrieval involves reactivating a memory trace. We saw that if we have the memory trace of somebody's face as a result of learning about them, we meet them and there's Joe's face and we make a memory of Joe's face in the face area. Well, we can reactivate that memory trace when we imagine Joe's face. So imagining a face is the reactivation of a memory trace and setting up loops with the working memory system. Likewise, we saw that hippocampal neuron that responded to The Simpsons initially, when the subject later recalled The Simpsons video, that neuron reactivated again. In fact, it reactivated just prior to the person reporting that they remembered The Simpsons video. So we were witnessing what retrieval is doing. When we retrieve a memory, we're reactivating the same neurons that were involved in processing the stimulus in the first place. Likewise, with the tiger situation, if somebody says, imagine a tiger, well, that will activate the concept of a tiger, which can also activate all linked memory traces, like what a tiger looks like and what a tiger sounds like. And a moment later, I'll be setting up loops of neural activity with the working memory system, and I'll be imagining a tiger. And so we can state the reactivation hypothesis is the idea that the same areas of the brain, for example, that are activated when we see a face are going to be involved in storing memories of faces that will then be reactivated when we retrieve the memory of a face, like during imagery, when we're imagining a face or recognizing a face. So the reactivation hypothesis is supported most directly by the hippocampal experiment, where the very same cell that was active during the perception of the Simpsons video was reactivated when the person was retrieving the memory of that Simpsons video. Memory traces can be localized, but a single learning experience can be processed by distinct neural pathways, forming distinct memory traces. And we can see this in the cortex and the hippocampus, for example. So if we're looking at a tiger, not only is the cortex making some weak memory traces, but at the same time, the hippocampus is making a strong memory trace, the episodic memory trace. So we've got memory traces being made in different locations simultaneously. We see this with the hippocampus and amygdala with the fear conditioning situation. At the same time, the hippocampus is making a memory trace of the context, the whole episode. The amygdala is making a memory trace of the specific association between the tone and the shock. Brain regions talk to each other and work together. So the visual cortex is sending information to the face area. But when the face area is activated, it also sends information back to the visual cortex. So a lot of the brain communication is sort of these loops of activity, local loops of activity, sort of feedback loops. Another example was the amygdala and the hippocampus. The two structures talk to each other. The hippocampus is sort of telling the amygdala to fear the training context when we're doing fear conditioning, and the amygdala is telling the hippocampus, well, this is an emotional experience, so make a strong episodic memory trace, not a weak one. Make a strong episodic memory trace for that event. Working memory is a short-term system used to manipulate information and search long-term memory. And we're going to put this box in the frontal cortex, or whatever we're sort of thinking about right now. That information, we can hold it in our working memory. We can manipulate that information if we're doing mental math, or we can just keep thinking about something for a long period of time. We are sustaining it in working memory. Whatever's in working memory has the potential to be stored in long-term memory by way of the hippocampal system. And whatever's in working memory can search long-term memory. If I have the word tree in my working memory, well, the concept tree will be activated in my semantic memory, and then other strongly linked concepts will be activated as well, and perhaps one of them might become the content of my working memory at time number 2. Loops of neural activity sustain working memory. So if I'm looking at the tree, well, the tree neurons are setting up loops with my working memory system, and I'm experiencing the tree. I'm conscious of the tree. If I shift my gaze to the cat, well, now the tree loops stop and the cat loops begin. And so at the next moment, I'll have a cat in my working memory. I'll be perceiving the cat. These loops are also initiated when we imagine things. In this case, we are internally, in a sense, activating the memory traces and setting up the loops, in this case to imagine a tiger. The speed of neural processing is fast, and much of it is unconscious. We saw that from the time of looking at a stimulus to the time of pushing, it's about 1/4 of a second. Now this picture is a little bit, we now know, misleading. It looks like there's just chains of neurons, one-way flow of information, but in reality, in lots of these areas, there are local loops of neural activity that are involved in processing information. But a lot of brain activity occurs unconsciously. We saw in the case of words, even if a word does not reach consciousness, there's early visual processing of that word. So the word, of course, is going to reach the visual cortex, and edge detecting neurons will detect the letters, the different edges that make up letters. Then the word form area, if we've seen the word before, we will activate a memory trace of that word. And all this early visual processing can happen without consciousness. Consciousness will require that this neural activity spread to the working memory system. Consciousness can be defined as the capacity to report a stimulus and can be studied experimentally. In fact, the binocular rivalry condition that we experienced in class, you put the glasses on and then you sort of see the face one moment, and then the next moment you see the house and then the face. This is a technique used to study consciousness. And what scientists have discovered is that when we're seeing the face, it's the face area that's turning on, and loops are established with the working memory system. Then it shifts to the house. Well, then it's the place area of the brain that is turned on, and those loops are engaging the working memory system, and it goes back and forth. It's like the two stimuli are competing for access to working memory, but the one that's in working memory is the one we're conscious of at that moment. We can also study consciousness using a masking procedure. You flash a stimulus and then a mask afterwards. Depending on the delay between the stimulus and the mask, you can either make the stimulus conscious or make it unconscious. Even though the stimulus might be presented on the screen for the same amount of time, it's the placement of the mask. When the mask is within 50 milliseconds of the stimulus, the mask blocks the consciousness of the stimulus. If the stimulus is a word, we see the different conditions here. The first one will be conscious. There's nothing to interfere with the loops of neural activity. The second one will be unconscious. The mask is within that time window. So when the visual cortex is setting up the loops to process the word tree, the mask comes along and interferes with those loops and prevents the activity spreading to the working memory system, so we're not conscious of tree. Here, the mask is timed so that it's outside the window, so the tree loops are allowed to happen. And in the fourth condition, we present the word tree for long enough that the loops have happened and spread to working memory, and it doesn't matter when the mask arrives. Consciousness requires the ignition of neural loops of activity that spread to working memory. Here we see fMRI, the difference between a conscious word and an unconscious word. Even for the unconscious word, we are getting visual cortex activation here. But notice you do not see activity in the front part of the brain or the parietal cortex. This is the working memory system. Whereas if the word reaches consciousness, we see this spreading activation. We can think of this then as the word is entering the working memory system. And working memory is characterized by activity in frontal cortex and parietal cortex, in certain regions within those areas. The binocular rivalry could be modeled here. When we're seeing the face, we're setting up the face loops, and then it's as if the brain is still registering the house. It's sort of like the house area is saying, hey, wait a second, it's our turn, and activation of the house area will then suppress the face loops, and the house loops will start up, and then we see the house, and then the face area is saying, okay, well, you've had your turn, now it's our turn, and they inhibit the house loops, and so then the face loops go again, and we just see the alternating face, house, face, house. Blocking the required loops will prevent a stimulus from becoming conscious. Again, if we see the word tree out here, the tree will be processed by the visual system in the word form area, and loops will be set up here, and kind of like a car ignition, the starter motor, the loops will be cranking here, and then it will get to a point where it can spread its neural activity to the working memory system, and the car starts up, and then we become conscious of the word tree. So for a piece of information to become conscious, it has to get up to this working memory system where then it can be shared with the rest of the brain. Then we experience consciousness of the tree. In the condition where the mask arrives just after the word tree, the mask seems to be arriving at the visual cortex at just the right moment that it can interfere with these loops, so we get no spreading to working memory, and therefore we're not conscious of the word tree. And finally, consciousness and working memory are closely associated. The function of working memory / consciousness is to share information with other brain regions. And it is still a mystery, however, why that particular pattern of brain activity is experienced consciously. After all, it's all just neurons. But when we have a certain pattern of neural activation, we experience it as conscious. Now, in the diagram here, let's say the word tree has entered my consciousness. Well, what the working memory system can do then is share that information with other brain regions. For example, with the hippocampus, then I can remember the experience of seeing the word tree. Or maybe I'm thinking about how to say what the word tree sounds like. I can activate that memory trace. Or the meaning of the tree, I can activate those memory traces. Or if I'm going to say the word tree, I can activate the motor programs to say the word tree, etc. So we want to think of then the contents of working memory and being conscious in general as a state of the brain where information is being shared with other brain regions. And this is a nice system. It means that the other parts of the brain do not all have to be interconnected with every other part of the brain, because you have this central clearinghouse of information. If whatever's in consciousness, it can be sent to other brain regions, and then those brain regions then can sort of contribute whatever specialized knowledge they have about the item, and that will help us make good decisions in the future. And so we can think of this consciousness that we enjoy as really just the tip of the iceberg, what's happening in the brain. If we're in some situation at time one, well, whatever's in the working memory, I'm processing this situation. I'm going to be activating memory traces and knowledge about the situation, and maybe I have to act in some way, and I've got two possibilities. I could do A or I could do B. Well, those two kinds of possibilities might enter my consciousness one after the other. And this would be when we're sort of deliberating. Should I do A or should I do B? But we experience it as just sort of, all right, no, I'll do A. No, I'll do B. And these items just come in a serial fashion, in a sequence. But what we don't experience are all of the unconscious processes happening in the brain. And these processes, if they're to reach consciousness, they have to be loaded up into the working memory system.